Thank you. I would like to thank the steering committee for inviting me to present today. And what I wanted to present briefly today is a little bit of my background, my research program here at UCF, and also introduce you all to the Genomics and Bioinformatics cluster, for which I am the lead. So just a quick bit about my background. So I joined UCF in 2016. And I do have experience both in the nonprofit sector and also in industry. In fact, I got my PhD in the last millennium from UPenn in computer science. And then, sort of the beginning of my career, I worked at Celera Genomics, where I was part of the Human Genome Sequencing Project, and then later at the J. Craig Benther Institute, where I was the director of informatics and a professor of informatics as well. And at Human Longevity, a startup company where I was senior director of bioinformatics and the microbiome lead. With regard to my research, while I started out in my graduate school working on approximation algorithms and theory computer science, I quickly moved into the area of computational biology and bioinformatics. And I've been working in the area of genomics, and in particular in the last 12 or 13 years or so in the area of trying to understand microbial communities and microbiomes, which is what I'll talk about a little bit today. My funding sources over the years has been both NIH, NSF, and also some of my projects have been funded through DOE and NIJ as well. In terms of research contributions over the years, as I mentioned earlier, early part of my career, I contributed to the human genome and mouse genome projects. So it was over 20 years ago. And more recently to the global ocean sampling expedition. This was a big effort to understand the microbial diversity in the world's oceans. And the techniques that we developed there were what we brought to bear to study the human microbiome, which NIH had funded a few years back and which we were part of as well. In terms of research and teaching here at UCF, I teach two courses, so one undergraduate and one graduate course. And my research group currently has 3 PhD students. One student just recently graduated. She's now doing a postdoc at UC San Francisco Medical School. And I do have students both from computer science and biomedical sciences as well, sort of an interdisciplinary group, if you will. So just a little bit more about the research, high-level sort of idea of the research that I conduct. And it's in this broad area of metagenomics and microbial community analysis. So microbes, those tiny organisms, invisible to the naked eye, but which constitute around half or more than half of the biomass on Earth, right? And they play such important roles in all aspects of Earth's biogeochemical processes. Many of these communities are host associated, including associated with humans. And so they have big impacts on the health of the host as well. And in terms of techniques, the fundamental paradigm of metagenomics, which really took off about 15 years ago, whereby you're able to collect samples, so be it water samples or stool samples or any other biological sample. And instead of trying to study microbes by growing them or cultivating them in the labs, the idea is to just extract the DNA, fragment it, and sequence it, and then use computational techniques to try to make sense of the microbial genomes and their functional capabilities. So metagenomics as a paradigm has been extremely useful and really has led to a great deal of progress in our understanding of life on Earth, in fact. And fundamentally, from a computational perspective, really what that boils down to is, once you have the sequence data, what you're interested in doing is really trying to understand, like I said earlier, the microbial community ecology and function, but there are a series of sort of steps that are involved. I mean, these DNA sequence data that you get back, because of this approach that we took for sample processing, you end up with fragments of DNA from microbes from all over the place. And so computational problems that involve a whole variety of questions, including trying to figure out the origins of these sequences, so the taxonomic assignment, trying to then reconstruct genomes of these organisms, then trying to figure out what are the genes on these organisms, and then using that inference to then figure out what we think might be the metabolic or the functional potential of these organisms. And then using that, so you can see sort of different levels of analysis, if you will. And all of these are, in a broad range. I mean, my group has worked on these problems over the years, involving a combination of sort of combinatorial algorithms and machine learning approaches. And we've really taken this to study different environments, including global oceans, air, and human environment as well. And I'll tell you a little bit more about our recent work on the gut microbiome, where, of course, the gut, we have, average human has about 30 trillion cells, human cells. And there are roughly as many number of microbial cells also in contact with our human body. And given that these microbes, have been around for a long time, you can imagine that they have important roles to play in our health. And indeed they do. And the last 10 years or so, I've made very clear some of these roles that the microbes play in our health. And some of my projects have been in that vein, in trying to understand, in the context of human health and disease, what are the microbes, that is these biomarkers, both in the context of sort of diagnostics, but also therapeutic perspective, or how we can use microbiome information to then inform us, understand about human diseases. I might add that in a computational context here, in this particular area, there is, in my mind, a big room for explainable machine learning approaches. And I'm happy to talk to my colleagues or any of you who have questions about this as well. My students and I more recently have been working on trying to use metagenomic sequence data to be able to infer microbial associations. So that is the whole idea being that, look, the abundances of organisms within communities that we're sampling are a reflection of their interactions. Now, the way we're assaying them are using sequencing. So from that, you can get what are called relative abundance data, compositional data. And from the compositional data, the idea is to then infer back these graphs or networks that can capture these associations. And these problems can be modeled as graphical model inference problems, sparse networks, and mixtures of sparse networks, if you will. And so we've been applying these techniques to really elucidate microbial ecology in different healthy human gut environments. So here's a figure with 4 different or networks in 4 different populations. Just at the high level, you can see that the structures look quite different. And we do understand that gut microbiomes are, of course, impacted quite a bit by things like diet and other factors. So we're using some of these observations then to really tease out what might be their impact in human health and disease. In terms of collaborations that I have ongoing and both near term as well as future planned, we have an ongoing NSF funded project with Dr. Molly Jewett studying Lyme disease and the microbe that's responsible for that. And with Dr. Sampath Parsastasi and Steve Ebert and Alex Cole as well. With Dr. Melu Sol, I have a microbiome-related project in understanding human health in the context of sort of mechanical ventilation events. And several other ongoing projects with folks at Vanderbilt and at Nemours as well. So the funding sources for many of these that we're looking into, so currently and planned, are mostly NIH related. So now I briefly want to introduce you all to the Genomics and Bioinformatics cluster, which is an interdisciplinary initiative that was started at UCF back in 2016 when I joined as a lead for this cluster. It was an initiative between across multiple colleges and departments. And the 4 other faculty members that were hired as part of the hiring effort are in CECS, College of Sciences, and also in COM. And now we also have secondary appointment from folks from College of Nursing as well. So it's truly an interdisciplinary effort. And our goal really here is to develop integrated interdisciplinary research programs, right, in genomics and computation and data sciences, that's really going to address some of these important scientific challenges in the life sciences. We also, of course, want to train and develop the next generation of research, sort of workforce for the life sciences domain. We have 15 faculty and research members who are currently associated with the cluster. We're also very pleased to have an NSF Career Award winner as well, Dr. Charissa Becker, who won it last year. We brought in, the faculty and researchers brought in 3.3 million last year. Broad, different research areas, if you will, including, again, we have folks working on computational aspects. We have folks working on things like biodiversity, conservation. Of course, myself and some of the other folks also work on microbiomes in the context of diseases, environmental conservation, and ecology as well. So I hope that gives you an idea of our cluster. We also have put together basic computing and infrastructure resources for the cluster that's also available to all faculty on campus. And in fact, all of our infrastructure, at least the sort of basic sequencing infrastructure in the labs, are all located in the biological sciences building as well. So with that, I would like to acknowledge Dr. Davi Reinert and Tamara Gabris and the FCI team for really helping the cluster as we grow. And of course, all of my colleagues in the cluster team and my research group currently. And also the chairs of the different departments. I mean, my chair, Dr. Gary Levins, the biology chair, Dr. Graham Worthy, Dr. Drift Parks from Burnett, and our dean, Dr. Georgiopoulos. Really, I would like to thank all of them for helping our cluster grow, and really kind of has they've all been very supportive of the cluster since the beginning. So I'll stop there and I'll take any questions.